All right, so here we are, the final installment in APHY 102, which covers the genetic component of how we become what we become in terms of our what we call our phenotype. That, our shape, our size, our skin color, eye color, what we call traits. Traits are passed through a phenomenon known as heredity. It's very common that people will talk about someone's kids as, well, you look just like your dad, or that kid's a spitting image of you, or your mom, or that's, or you have your mother's eyes, that sort of a thing. So for a long time, people have wondered how this information is somehow transmitted that leads to people looking like people whom they're related to. Um, the and we just have a few of the basic ideas. Again, more about how to wrap your brain around what we now know of as the heritable material. The original idea that was for a long time was called the seed idea, which is where a man plants a seed in a woman. That it's also known as the homunculus. A homunculus is a small human being. And so the seed idea is like this picture over here on the right, where you have a small human being stored inside of the sperm of the man, and it's planted into the female. Then there are some ideas about blending hypothesis, that somehow there's some genetic material that then can be mixed. And so the child is somewhat of an in between. Now, that does have some merit if you think about how you have partial dominance and so forth. So there is some blending aspect to this, but it doesn't explain everything. Then there's this panspermia, this idea that there are particles that somehow emanate from like your pinkies and your toes, and somehow they affect how a person's offspring looks. And then lastly, this particulate hypothesis. Traits are based on discrete heritable units, factors. Again, not unlike blending. All right, each of these have some aspect that get into the modern understanding of heritability. This idea that we do inherit things from our parents. They blend in some cases. Yes, we are implanting material from the man into the woman. And there are aspects of panspermia, that is, that we do have some segregation and so forth going on of traits. In the grand scheme, given how much we understand molecular genetics now, the fact that we really have only understood it for the last 100 or 150 years is actually quite shocking. I mean, that is shockingly recent in the large span of observation of human reproduction. I mean, we've been studying reproduction for thousands of years, back to the Greeks and even before then. So that 150 years is like the last period on the last sentence of the history of understanding of genetics. But even though he came up with some of these things, those were just hypotheses. We call them laws now because they've been corroborated in multiple ways for decades. But back then, they were just hypotheses, and it took 40 years. So it wasn't even until the 20th century that we began understanding the implications of Gregor Mendel's work. So we call it the law of dominance. That is to say that there are traits that will override other forms of traits. So we call that a dominant or a recessive in some cases, and now we consider that a law. Then there's this segregation. Again, when Gregor Mendel came up with it, it was a hypothesis, but now that there has been some additional experiments that are independent and corroborate, now call it the law of segregation, that genes can move in independent ways from parents to offspring. And they assort independently. That is, they are discrete elements. All right. So he's the father. He came up with the original concepts, and they have then been proven and corroborated again. So now that we have what we call laws, we have not found any refutation of these three major laws. So now that we have some of the history and the basics of what's going on, we're going to now get into the mechanisms that we now understand that reinforce what Gregor Mendel observed with his pea plants? Well, we have genes, which are functional units within the DNA. Turns out that a surprisingly little amount of our DNA is actually genetically active. That is to say, encodes proteins. All right. So a gene typically encodes one or more proteins, and it really comprises only a relatively small amount of the overall DNA in a chromosome. So a chromosome is a collection of genes in DNA strand. Then we have multiple chromosomes that give us what we call the genome. So we have 22 what are called autosomes, and then one more pair. So we have a total of 23 pairs. That other pair are called sex chromosomes, which are X and Y. So we have a total of 46. We inherit 22 from our mothers. We inherit 22 from our fathers. Those pairs, the reason we call them pairs is that they have almost always DNA that encodes the same genes in the same locations on the chromosomes. So that's what they means. They are homologs. All right. So the gene that encodes brown eyes can be found on the same location on the same chromosome, both from the mother and the father. We have, and this number bounces up and down depending on how we look at it. It used to be higher, it used to be north of 30,000, but now we're finding out that a gene can encode more than one kind of protein, so that number has actually dropped a little bit. And the sequence of the DNA, there are 4 nucleotides, gives rise to the information that gives rise to a 3-dimensional protein. So if the nucleus is sort of the library, if you will, of where genetic information that encodes proteins is stored, how do we manipulate which genes are turned on and which genes are turned off? We don't express every gene in every cell, which is why a muscle cell looks different from a neuron, which looks different from an endocrine cell, which looks different from a skin cell, and so forth. Each cell only expresses a subset of the total genome, even though they contain all of the genes. Well, that's where control can occur, and control is the result of signaling. So for instance, maybe we have a signal from a cell, it could be like a hormone or other types of signals that then lead to events in the cytosol that we call signal transduction. So we're turning whatever the original signal into the language of the cellular biochemistry. And that may activate what we call a transcription factor that goes into the nucleus and determines whether or not a gene is turned on and how turned on it is. Maybe we want to turn on a gene maximally and express lots and lots of that protein. So we would turn it on and we'd make a lot of mRNA. That mRNA is then moved out into the cytosol where it is translated into a 3-dimensional protein that can now affect some kind of a response in that cell. So things like hormones can do this, cytokines, which are hormone-like molecules, or perhaps contact. There are certain types of mechanical stimuli. It can be temperature, hot or cold. Lots of things can drive changes in genetic expression. For instance, there's really cool genes called heat shock proteins. So when the cell gets too hot, it'll turn on transcription factors that then will encode proteins that protect the cell from being a little too warm, and cold shock proteins for that matter. However, if we have issues with the genetic material, like maybe there's a gene that encodes a protein that may cause disease, then we might have transmission of that disease down through generations. So, like for instance, we see the father here and a mother that is not affected. The father seems to be affected, and then some of the kids. They had 3 kids, 2 of which have the disorder or the condition or the trait. And one of them marries and has 3 kids, and again, 2 of those kids have that condition or trait. That's these things called pedigrees, where we can follow whether or not somebody has a given trait or condition. Gives us the ability to predict risk in the case if it's a disease, like for instance Huntington's disease or cancer or sickle cell anemia, diabetes in some cases. We can begin to assess risk probability and maybe intervene earlier in that patient's lifetime in order to mitigate the circumstances of that having that gene. BRCA1 in cancer is a classic example of that. If you have a mutated BRCA1, you can't repair your DNA as effectively, which means you're more likely to suffer from certainly breast cancer, but also other types of cancers. And cancers do run in families, so you can do some risk assessment, and maybe that merits a different therapy than somebody who is not in a pedigree where you have a commonality across generations. One way we can establish pedigrees, and one way that we can determine if a child has a certain gene or that's a risk factor, we call those chromosomal tests. And the two that are classic are amniocentesis, where you insert a needle to draw off amniotic fluid, or in other cases you can take a sample of the chorion, the maternal side of the placenta. You can take a sample and do it that way. We're going to talk about a newer test here in a second or two. So those are the traditional ones. They do have a little bit of risk associated with them, but depending on the severity of the disease you're talking about, that risk may be worth taking. So we can see here, this is just an example of a pedigree where we have an affected person in red, and we have unaffected in blue. And it's not just about whether or not you have the condition, but also whether or not you can pass on the possibility of having that condition. We call unaffected patients, depending on the type of dominant or recessive gene, we'll talk about that in a little bit, can be carriers. Cystic fibrosis is a classic example of this, where you can be a carrier, and if two carrier parents have children, then they can have children who are affected by cystic fibrosis while they themselves are unaffected. One way we can analyze for potential disorders using amniocentesis and so forth is what we call a karyotype. That is, you isolate the nuclei of the offspring or the person. It can be an adult; it doesn't have to be a child. And you look for dividing cells that have condensed chromosomes, and then somebody can sort the chromosomes by their pair number. And people get really, really good at it because these look, you can't really see here, but chromosomes do have a specific type of stripe pattern. And so folks that do this sort of work get really, really good at seeing the stripes and recognizing, oh, that's this or that's that kind of a thing. So here's a normal or a relative, certainly a karyotypically normal person. This person happens to be male, all right, because they have an X and a Y. At least genetically, they're male. Over here, what condition that we have? You look here, we're supposed to have pairs, right? So what's going on right there? We'd have 3 of chromosome 21. That's called trisomy 21 and is the most common cause of Down syndrome. One of the most common genetic aberrations that we see is trisomy 21, which gives rise to Down syndrome, 1 out of every 700 children. And here we have another karyotype. Let's diagnose. So we see homologous pairs, looking like it's working. All right. Oh, hold on here. We don't have a pair anymore. We now have 3 X's. That would be trisomy X. So in that previous patient, I'm going to slide forward here. What we have is 47 chromosomes, 3 X's. That makes it genetically female. Usually we have developmental delays. For whatever reason, in mammals and in animals more broadly, if you have polyploidy like that, it leads to problems for reasons we don't fully understand yet, because in plants can have polyploidy all day long, and it actually confers some benefit in some cases. All right. So here are just some common anomalies, like for instance, maybe they only have an X, they don't have two X's. All right, it leads to infertility and a short stature. There's not enough genetic effect from that one X to make up the difference for the other one. Klinefelter syndrome, I find that quite interesting, because are they genetically male? Are they a female? XXY? Well, oftentimes they will have morphologic maleness. The Y chromosome is exerting its effect, probably the SRY gene among others, leading to a more of a male morphology. But in any case, with that double X there, you might have excess female hormones, and so they have low testosterone and often are infertile. Already talked about triple X. You also have XYY syndrome. Again, because of the male genes on the Y, you overcome the default female body type. Females are the default pathway in humans. They might have tall stature, but they do sometimes have neurologic difficulties and developmental delays cognitively. And then we have things like mosaicism. And what do we mean by that? It means that genes from the Y may have made their way onto the X and vice versa. We do see movement of genetic material from chromosome to chromosome. It does happen. And you see it frequently in certain types of cancers. All right. So that mechanism of transposition of genes is a thing. And so what we get is maybe we have a 46XY, but now they're ambiguous in terms of their morphology. Even though they're XY, they may have female body morphology. They may have testes where their ovaries are. They're 45 situation where they're missing an X, but yet maybe they have maleness genes on that X and so forth. And so you do get some conflict in terms of maleness and femaleness. As much as the pundits would want to say you're either male or you're female, that's just not true, and it's heartbreakingly sad that we need to dumb down our understanding of human male female identity and or morphology. XX males are classic of this. They have XX, but they are in fact morphologically and biologically male. Okay, that's probably due to the fact that that X or that Y genes have transposed onto the X chromosome that the child inherited, and so therefore there's SRY or maybe some other maleness-oriented genes on that other X that then drive a more of an XY phenotype. Some basic causes of these anomalies that we see, or these genetic deviations from the 46, 45 plus 1, 22 pairs plus the sex pair, sometimes when during cell division, during mitosis, the chromosomes get tangled, and what we get is a failure of disjunction. That is to say, the chromosomes don't separate cleanly, and so pieces of the chromosome may leave on one, or you're missing pieces of the chromosome, or you get extra chromosomes. That's where you get like trisomy 21 and that sort of thing. Maybe there's a change in the base sequence, and that can be due to exposure to what we call mutagens. Even sunlight is a form of mutagen. Inflammation is actually a mutagenic phenomenon because it drives cell division. The more cells divide, the more prone they are to having changes in the DNA within the genes. Parental influence, and I'm actually incredibly intrigued by this. This says that there may be changes to the chromosome that are not related to the base pairing sequences. That is to say, there are aspects of DNA that can turn genes on and turn genes off that have nothing to do with genetic sequence. And that's why they call it epigenetic. That means it is above the gene. So we know for a fact that that parents, not just mothers, but parents that undergo extraordinary amounts of stress during their reproductive years, we will see effects in those children. All right. And they are not due to the base sequence, they are due to how the DNA is modified in other ways that have nothing to do with the base sequence. So that's called epigenetic. You can also have errors that may not be occurred with cell division. They may just occur spontaneously or randomly, or perhaps due to perhaps exposure to certain types of chemicals and so forth. And so we get duplications or we get deletions in some cases. Turns out that as we age, our chromosomes change a little bit. We're losing material at the end of our chromosomes. Now, there are enzymes that will help to stave off that. That's part of the aging research, is that we are losing little bits of chromosomes, but there are enzymes at the ends of those chromosomes to try to keep that from happening. But eventually, the loss overcomes that, and you get deletions going on, and that leads to aging a little bit. Or you get accidental duplications and so forth. Just it's a mutagenic process. And advanced parental age is considered a risk. The older the parents are, the more likely you do have chromosomal abnormalities, and so forth. And mothers, generally age out sometime, in their 40s or late 40s or early 50s for first half of their age 50. Some go longer. Some go into their 60s before menopause. Guys can usually be fertile well into their 70s. It's unusual, but it does happen. But in either case, if the parents are of elevated age, there are risks. And exposure, as I mentioned, exposure to mutagens. That can be radiation, and it doesn't have to be like nuclear radiation. It could be like, for instance, cosmic radiation. Turns out that people that work in the airline industry on aircraft are exposed to elevated levels of cosmic radiation, and the longer they fly and the more time they spend in airplanes, or astronauts go out into space, there's you lose the shielding of some of the atmosphere, and so they have elevated risk. Occupational exposure or environmental exposure to things like heavy metals in some cases, or benzene is a known mutagenic, and that's a primary component of gasoline. So if you happen to live near an oil refinery or around the corner from a gas station, groundwater may have a little bit of benzene in it. And again, we talked about disorders of sexual development. That's just one potential consequence of these chromosomal anomalies, sexual development disorders. But if you have a gene for it, there is a possibility that that something can happen to that gene through one of these mechanisms. Here is an illustration of amniocentesis, where you insert a needle through the abdominal wall of the mother, far enough to penetrate the amniotic sac and extract a little bit of the amniotic fluid, which will be rich in the child's genetic material, so you can do a karyotype like that. So you will isolate the cells and then look for cells that have the chromosomes that are identifiable, and you do a karyotype that way. Or you can obtain it from the chorionic villi. Same story. You're obtaining some tissue, you isolate the cells, and then you look for cells that have condensed chromosomes in order to do the karyotyping. There are more modern, less risky. Sometimes these are necessary for certain reasons. I'm not going to go into those. Part of the problem with these is that you do have the risk of inducing a miscarriage at about 1%, 1 in 100, pretty low, but not insignificant. So the most common test nowadays, because it's much safer And because the molecular biology is getting so much stronger, it's what we call non-invasive prenatal testing. Uh, what we have learned is that during pregnancy, the fetus will actually shed DNA that will move into the mother, and even entire cells. It's been recently discovered that there are actual cells, particularly involved in immune function, that move out of the embryo or the fetus and actually supplement and assist the woman's immune system. It's actually extraordinary that way. But the other benefit is that you have the ability to obtain DNA without having to do an amniocentesis. Sometimes you still have to do the amniocentesis, but for about half a dozen or so tests, you can now do this NIPT. Now, it's possible that I used some of these terms earlier, and I would just want to make sure we have these for you in any case. So when we say genotype, this is the type, the particular sequence gene that you might have, and you can usually only have two possible genotypes for any given gene, because if you have two different genotypes, one from the mother, one from the father. So genotypes are variances in the gene sequence and so forth. So we have two genes, and then those genes, depending on their sequence, can confer traits. We often call traits phenotype. That's the physical manifestation of the gene effect. Right? You can't always know the genotype based on the phenotype. So the manifestation may be determined by other factors, or there may be more than one genotype that confers a given phenotype. But we don't have to go down that rabbit hole all that much. So we have the genetic sequence up here. We have the manifestation here. The genome, on the other hand, is the collection of all the genes in either an individual, or you can even talk about it in terms of species. So a species level genome would show you all of the different genotypes for all the different genes. And we've also mentioned homologous, that is to say, we can spot chromosomes by their shape, their size, and their banding patterns. And chromosome 1 from the mother generally looks like chromosome 1 from the father, and so forth and so forth. And it takes a little bit of practice, but people can get there. So even though the chromosomes generally look the same, the genes on those chromosomes may vary. So we call those individual ones from mother and father, sometimes refer to that as alleles. So here's the definition of gene. We've already talked about it. It's the one that contains the information, the coding sequence for making a protein. So we should have two genes of each, or two genes, one from the mother, one from the father. The only variance in that is XY most of the time, because those are not homologous chromosomes, they're sex chromosomes. Like genotype, another way for saying that is allele. Okay. This schematic right here shows you the process of meiosis. That is, how do we take a parental cell and create a gamete? So we have one parental cell and we make four gametes. This is true with sperm. In eggs, you do have the haploid cells, but three of the four are not gametes because you're condensing the cytosol into one. So these become polar bodies. But in any case, there are two separate divisional events. The first event, what happens is, it's called meiosis 1. You duplicate the DNA in each of these chromosomes. So they're a single strand here for now, and now they become duplicated. Now we have two sister chromatids on one chromosome. This one only has one quote unquote chromatid. It's a chromosome in this form here, but it duplicates. Same thing with the other one over here, and we separate those homologous chromosomes into new cells. So that's sort of a reductional event in the first divisional phase. So now we only have half the original genetic material that we had from in terms of number of genes from the parent. Now this doublet here will then separate into individuals, and so now you have true haploids here at the bottom. So there's a divisional one that's reductional, so you now have half the overall genetic sequence in each of these cells. And then what we do is we split those sister chromatids during the second division. So we're reducing to haploid in the first division, and then we're dividing the sister chromatids in the second division. It would be nice that we could say that each gene has a given phenotype, and that would be lovely. But unfortunately, it's not that simple, and probably with good reason, because nuance and in between are part of variation, and variation improves the flexibility of a species. And so some genes are able to manifest a trait more readily than others. And so the ones that can manifest a trait we call dominant. Ones that can only manifest a trait if you happen to have two copies of that gene are called recessive. So let's look here for a moment. Here the seeds are yellow because that individual happens to have two yellow genes, one from each parent. But if one parent has a green gene and the other one has a yellow, it's still yellow. That's because whatever is made by the yellow overcomes whatever is made by the green. Now, if some of the offspring may have green seeds, that's because they inherited two greens. So the greens are called recessive. And lots of disorders that we see in clinics are often referred to as autosomal recessive. That is to say, they're on non-sex chromosomes and they inherited two recessive copies of the gene, which lead to the condition. Cystic fibrosis is that way. And once Gregor Mendel figured out this idea of dominance, he was actually in a brilliant way able to determine the probability of traits. And this is known as a Punnett square, and I've got one filled out here. And what you do is you put the genotypes for the mother down the vertical axis, and you put the genotype for the father on the horizontal axis, or you can flip them. It doesn't really matter that much. And then what you do is you insert the letter in the square. So we have a capital B here and a capital B here, so we have BB. Capital B here, lowercase b here, and so we have a mixed genotype here. Lowercase b, and we always put the dominant allele first. So even though this is on the left side, it goes second, but because we had a capital B from this parent, so we have the mixed genotype here, and now we have two recessive copies. And so what we see, if these parents were to have a number of kids, more than four, because sometimes you kind of buck the probabilities. If they had, let's say they had 1,000 kids, what we would see is that we would see a 3-to-1 ratio of phenotype, because we would have dominance here, dominance here, dominance here, but recessive here. This is a little complicated, so we're going to walk through this slowly. Before we had a full understanding of genes and phenotype, the word that we used for the heritable factors was called alleles. We now say genotype, right, or something along those lines. What we have found is that these alleles, if you will, the mother's and father's genes, do not run together. They don't normally merge. You don't see in betweens unless you have what's called codominance, but we're going to get there eventually. Each gamete carries only one copy of a gene, so only one allele at a specific site, location. So a location, or in Latin locus, All right. So we know that a given gene can be found at a certain location on a chromosome. That's what this is saying here. And so you should have only one copy of that gene, one allele, and you inherit an allele from each parent. And then you get into dominance. But it's law of segregation. So let's say if you're developing gametes, what you're going to see is that you're going to get one copy of the gene or the other in those gamete cells. All right. So what we've found is that when we look at meiosis, this is where that segregation actually occurs. So Mendel observed the phenomenon, but he didn't really have a handle on the genetic aspect of it. He just saw the properties in his beans. And once we understood meiosis and what was going on in meiosis, that corroborated the observations that Mendel had. In our first Punnett square, we were looking at one gene, all right, actually two, but looking at one individual gene and the parental copies of those. You can actually do what's called a dihybrid cross, where you have multiple genes. This is actually two in this case. And you're looking at the genotypes for this S gene and this Y gene, and you can do this all together. Now, we don't have to know how to do this dihybrid cross. It's not as hard as it looks, but it does get kind of complicated. And by being able to figure this out, Mendel was able to identify that the S and the Y do not run together. That is to say, they separate independently. You can have any number of combinations of S and Y. So you can see here with this capital S, lowercase s, Y, capital Y, lowercase y, capital S, lowercase s, capital Y, lowercase y. So the possible combinations are 4. All right, they separate independently of each other. This independent assortment of alleles and separation and so forth are the mechanism that describe what I told you in class a couple of days ago, which is the point of sexual reproduction is diversity, diversity, diversity. And so we have 20,000 genes, of which we generally have one copy from each parent. They separate mostly independently of each other, and that separate and divisional and so forth means that we can have any number of combinations in the offspring. And that diversity then may account for, well, maybe a gene confers some kind of a benefit that allows survival or thriving in the environment, or maybe there's another gene that might have been beneficial at one time but isn't beneficial now, maybe a hindrance. And so by these combinations, you can achieve an extraordinary amount of diversity through sexual reproduction. And while Mendel's discoveries are considered some of the great intellectual leaps in understanding, nevertheless, he didn't get it entirely right. He was using a relatively simple system of P's. And yes, he did see some variances in his numbers, but he kind of stuck to his guns a little bit, which was good in terms of being able to identify some basic ideas, but it doesn't explain some of the more complicated phenomena. And these more complicated phenomena we now call non-Mendelian because they don't obey the same frequency and up or down kind of mentality that Mendel had. So some of these exceptions we're just going to kind of just briefly explain here, just give you a basic definition. Codominance, and we'll show this in the next slide. So I'll show you an example of codominance where maybe you have sort of a 50-50. So both genes are having an effect and they both lead to a mixed phenotype. Then we have polygenic inheritance. So maybe a given trait is not just controlled by one gene. And we see this everywhere. Like, for instance, diabetes, there's over 100 genes that are known to be involved in type 2 diabetes. If there was ever polygenic inheritance, that's an example. So it's not just one gene, one trait. It may be multiple genes influencing one trait, like body weight, for instance. Epigenetics, again, I obsess about this a little bit because I believe it serves as the basis for some of the health disparities that we see, due to the lingering effects of economic and social disparities. That is, if you have a population within a society that's undergoing a lot of stress, you may begin to see untoward effects in terms of health outcomes or incidence of disease and a pathology that may not be explained by the genetic sequences, explained by the circumstances that the parents have grown up or lived in. Gregor Mendel didn't understand the idea of sex-linked because plants can sort of split the difference on this. They can be both male and female. You do have male and female plants, but some plants are both. So he didn't get a good handle on that. And now that we understand that there are male and female chromosomes, now we have a better understanding of, like, for instance, red-green color blindness is almost exclusively in males because the red-green trait of being able to see or not see red and green resides on an X chromosome. So males only have one X chromosome. So if they get one inactivated X, then they're going to be red-green colorblind. And then lastly, incomplete dominance. Along with codominance, we will show you what we're talking about in the next slide. I can't explain it properly, so let me show you the image in the next slide. So I think this figure illustrates well the differences between codominance and incomplete dominance. So in codominance, you're expressing the trait, but in a mixed way, not a blended way, but an all or none mixed way, like the spots. Some of the spots are orange. Actually, their word is rufous. All right. And some of the body is white. So you have this combination of rufous and white in the children. That's called codominance. They're both exerting a dominant trait. It's just that they're sort of dividing the real estate, if you will. Incomplete dominance is well shown here in this flower, where if you inherit a red gene and a white gene, what you get is this pink intermediate. All right. What we see in terms of certain types of diseases is that if you don't have two functional copies, you may have a little bit of a deficit in the trait, which can lead to disorders. In this case, certainly it's not a bad thing to be pink normally. Perhaps you need to be red in order to attract hummingbirds or something. But this is incomplete, and this is codominance. In humans, codominance can be expressed as a form of blood type. Both A and B are dominant alleles. And so if you inherit an A and you inherit a B, you'll be AB. All right. And you can look here. So we have an AB parent and an AB parent. So 25% approximately of the offspring will be just pure A because they inherited an A from both parents. 50% will be that mixed phenotype, because you're going to either inherit an A from one parent and a B from another, or the A from the other and B from the one. And then finally, 25% will be pure B, having inherited the B from both parents. So this is a clear example of codominance. So here's the definition of polygenic inheritance for you. A great example is skin color. You can be very light, very dark, and every shade in between, because there are multiple genes that are affecting skin color. And the same thing true with human height as well. So here are various examples of different skin colors. Again, it's not It's not even one or the other. There's in-betweens and even subtle shades of in-betweens. That's because we have multiple genes that can manifest in the skin that give the skin its individual colors. And this picture is kind of cool. What we've done is we've taken a population of roughly, oh, and I want to say here, a little more than 100 individuals, and we have sorted them by height. And you see that we get a sort of frequency distribution, that we have people that are relatively short, people that are relatively tall, and you can see the frequency. For instance, I know that I'm 6'3", so that's about the 95th percentile right here. So of the 100 people here, you can see 1, 2, 3, 4, 5. So there's about 95 people on the left side of that division and 5 people on the right. The thing about polygenic inheritance is that given that we only can have 2 copies of a given gene in an individual, that's not enough to explain the variance that we see. So multiple variants of multiple genes can exist within a population. ABO is an example of this. It's codominant and polygenic. So we can get multiple genes, epigenetics, all driving phenotype. So environment plays a role in gene expression. And it's not always epigenetic. It may be that, if you live in a hot environment, you're going to express more heat shock proteins and that sort of a thing. So there's a wide variety. You may express genes in order to meet the needs of the environment, or it's possible that you can affect the genes by an environment. The 2 major epigenetic changes that we know about are called acetylation and methylation. And acetylation, when the gene is acetylated, it means that it is more likely to be expressed. If it's methylated, it is more likely to be silenced. So you have this sort of, turn on or turn off, and it passes down through inheritance. It's not the genetic sequence, it's the acetylation and methylation patterns that occur due to environmental interactions. A classic example of an environmental effect is sun tanning. All right. You can literally change your phenotype by exposing your skin to ultraviolet radiation. And so skin color can and does change based on your environmental interactions. Same thing with seasonal changes in fur. So coat color will change based on the season, and that's often due to light-dark cycles and also temperature as well. Over here, we have a pH-sensitive phenomenon. So if you have acidic soil, maybe you'll have a different colored flower. Even though it's the same genes, the gene expression has changed due to environmental effects. And as I described previously, sex-linked traits involve the sex chromosome. So in particular, red-green color blindness is on the X chromosome. And so the probability that a female would inherit the colorblind, red-green colorblind phenotype is significantly lower than the male. And in the classic sense, if a female is going to be colorblind in the red-green, that female would have had to inherited a colorblind allele from the father. So the father would have been by default colorblind, and a carrier mother. Maybe the mother was colorblind, maybe she wasn't, because maybe she had a good gene that was dominant over the recessive color blindness allele. So you would definitely have to have a colorblind father and either a colorblind mother or a carrier mother, one who has 2 different genes, and you happen to inherit the one that doesn't allow for red-green vision. And here is the Punnett square for this. If the father is not colorblind, he may be he certainly has a good X. He has to have a good X if he's not colorblind. The mother may not be colorblind, but she may be a carrier. And if she's a carrier, 3 of the 4, 75% of the offspring will be able to see red-green, but 25%, and only in male offspring would you see the red-green color blindness. Here we have a list of a term that I used earlier. What we mean by autosomal means that it is not a sex chromosome, all right? But the genes that would encode for a disease phenotype are recessive. So if you inherit a non-disease gene from one of your parents, then you will be a carrier, but you will not necessarily manifest the disease. Sickle cell anemia is along those lines, but it's a little more complicated because it is an incomplete situation. If you have some of those sickling hemoglobins, they will still sickle even though you have a lot of normal hemoglobin, and so the sickle cell anemia will not be as severe as somebody who inherited a full-on recessive situation. Cystic fibrosis is classic autosomal recessive because if you have a non-cystic allele, you will not manifest the disease. This one's actually quite a marriage breaker. Sometimes people don't even realize they're carriers until they start having kids, and then those kids pass away in their late teens and early 20s. And now we're much better at managing cystic fibrosis, but if while I was growing up, the typical lifespan of an individual was in the early 20s. And so in fact, my in-law family had experienced a divorce because of this phenomenon. PKU, also known as phenylketonuria, Tay-Sachs disease, which is another type of hemoglobin disorder. They are all examples of what we're called autosomal recessive. Why are they still in the human genome? Because we have carriers who can survive and thrive even though they might have that gene. In politics and in social discourse, a lot of people do not really understand this concept. They always say survival of the fittest. Well, they imply that you're in good shape and you're strong and you can, do whatever. The actual truth, when they say fittest, that means that you have the array of traits that will allow you to be successful. Maybe it's intelligence, maybe it's being one-handed or another, and so forth. So it's a combination. So natural selection involves certainly the transmission of genes from parents, the variation that's achieved in that genetic transmission, and then does that combination of genes provide a benefit or not a benefit? If it provides a benefit, then that individual is likely to survive, thrive, and reproduce. That's what natural selection. It's not about being stronger or faster and so forth. It is about the combination. So we're going to transmit genetic material and we're going to mix and match. All right. That's what sexual reproduction is about. And that new combination may have potential consequences that benefit or harm. Right. Natural selection will not happen unless we have all 3 of these things going on. You can have genetic transmission and variation, but if there's no consequence, then negative genes that could be a problem somewhere will still reside in the gene pool. If you get a heterozygote, that is, say, one gene copy that's different from another, maybe there are positive consequences. If the recessive allele is there and you get both recessive alleles, maybe that's a disadvantage. So we call this idea that if you have 2 different alleles, call that our heterozygote advantage, that the homozygote, two copies of the same gene, may not be as advantageous as a heterozygote. Here are the molecular mechanisms that help describe independent assortment and lead to variation in our gene pool. Some of these are very, very slow and only occur at certain times, and other times there may be extreme conditions that drive the enrichment of a gene in a given population. Let me give you sort of an example of this before I break these mechanisms down. Back in the 1000s or so, what we call the medieval times, there was an outbreak of a disease known as bubonic plague, and it wiped out a huge number of people. Literally, like 70% of the European population succumbed to it. And what it happened was, if we examine the genomes, the genetic material of people who succumbed to bubonic plague and those who survived, what we found was that there was a genotype that conferred an advantage in the bubonic plague. And so the people who were susceptible were killed off, and that led to an enrichment of the survival genes in the human population. So what are these mechanisms now? So we have mutation, and this can occur randomly or can be accelerated by exposure and other environmental consequences. We do get just sort of a baseline number of changes when the DNA is replicated. It's very, very low, but it does happen from time to time. During meiosis, and we haven't really gone into detail in meiosis, right at the very beginning, when you have both chromosomes from both parents, there is an event where the chromosomes will line up, homologous chromosomes will line up next to each other, and they will literally cross over and swap genes. So you end up with a chimera chromosome that has some of your mother's genetic material and some of your father's. So there's a crossing over. We call this synapsis. That will lead to that sort of independent assortment. That is, we have you may get a mother's chromosome in one division and a father's chromosome in another, and so forth. So we have all of these things are contributing to diversity. And maybe just one gamete out of hundreds of millions is going to fertilize that one egg that happens to be present. So these mechanisms all drive variation in sexual reproduction. We've talked about autosomal recessive. Now let's talk about a rather unusual example, and this is called a deleterious dominant. That is to say, if you inherit the dominant gene, you are likely to get the disease. And Huntington's is a classic example of this. So here's the question. So we have a 30-year-old here whose father died of Huntington's. Mother and sister do not have the symptoms. So what are his chances? You could do a Punnett square is what you would want to do. And the Punnett square would show you that roughly 50% is, so if you are the child of someone with a deleterious dominant allele, you have about a 50-50 chance of coming down. Now, why don't these things go away? Well, the truth is, if it's a deleterious dominant that affects early in life, they'll never get to reproductive age and that gene just gets lost. Okay. But what happens with these deleterious dominants, and this is why they're very rare, the key is that in the case, like, let's just go through this. In the case of Huntington's, it's a progressive disorder which usually doesn't claim the patient until they're in their until they're well into their 40s. The symptoms may manifest at age 35, well into the reproductive years. So they could have plenty of kids by the time they're 35, and then you start to see the symptoms in the parent, and then they might be dead by 45 or so. Huntington's in some ways reminds me a little bit of Lou Gehrig's disease, ALS, but it's not as uniquely motor. It's a neurodegenerative disorder, but it involves behavior, loss of the ability to communicate, and so forth. But it does have some pathological similarity to ALS. I just happened to put a Punnett square here so that you can kind of run through the situation in the previous case study. What you would have is that if he has a sibling that does not have the disease, that means that the father must have had a recessive gene. So he would be capital H, little h on one of these. Let's put him on the top, capital H, little h here. Now the mother doesn't have it, so she's got to be recessive for both genes, H, H. But that capital H there, the first, would manifest in this child and manifest in this child, so that's why it's 50%. Okay. Try it out. Just put capital H here, little h here, little h, little h. You have to have the recessives in order to not have the disease. Just a quick word about biotechnology. There's not a ton we're going to go into here, but understand this is where your career is going. That the therapies are increasingly molecular and increasingly about dealing with genotypes in order to influence phenotypes. Look no further than mRNA vaccines. So rather than giving even a heat-killed virus or that sort of a thing, you can give the mRNA to express the antigen to create antibodies. So it's coming. It's going to be a part of our therapeutic environment. So having some awareness about it is super important. CRISPR-Cas9 is the ability to target specific changes in genes. I'll give you an example. So they can now cure sickle cell anemia in patients who have sickle cell. What they do is they give them some chemicals in order to isolate the stem cells from the bone marrow. Then they apply this CRISPR-Cas9 to turn off the adult hemoglobin. So all they do is just change the DNA there so that you no longer express the sickling hemoglobin. And the fetal hemoglobin can take over. So they've learned how to edit it just right so that you can have the non-sickling fetal hemoglobin, and that alleviates the sickling condition. mRNA vaccines, as I explained. You inject the mRNA creates a protein. That protein then allows you to develop immunity without ever having been exposed to the pathogen. Very, very powerful. And you can change this quite quickly. That's one of the real advantages here. You don't have to grow up new cells and new things along those lines. All you got to do is change that sequence and off you go. And this has huge potential for things like malaria, for cancer, and so forth. CAR T cells, these are special cells that you can use in certain types of blood cancers. What it does is it helps generate it helps the patient generate antibodies against cancer, blood-borne cancer cells. Stem cell use, we've already talked about that with the CRISPR-Cas9. What we can do is we can use stem cells either from the patient, or we can use donor stem cells in order to replace bone marrow in patients. Like, for instance, maybe you have a certain types of bone cancer that you have to basically kill off the bone marrow. Well, then you can use bone marrow from an appropriate donor to replace. But you can also use these stem cells for diagnostic and research purposes. And GMOs. A classic example of a GMO is a mouse that now expresses a green fluorescent protein that comes from a jellyfish. and you can target it to different organs, and so you can see those organs grow green, or you can make the whole mouse green. Lots of possibilities out there. One of the really cool GMOs that are out there is that you can incorporate vaccines into the item in question. Like, for instance, if vaccines require refrigeration, you can create a genetically modified potato that can introduce the antigen so that you develop antibodies to that antigen in the potato, which doesn't require refrigeration. Lots of possibilities here. Another sort of on the side of this, for instance, people who are connoisseurs of marijuana, there are molecular biologists out there who have learned how to modify a marijuana plant in order to express more THC. The Canadians were particularly good at doing this back in the early 1990s. They were able to genetically modify marijuana to have a higher THC content. And so that does it. And in this case, we have a phenotype here. He's asking why a long face. It's because he had a genotype for a long face. All right. So enjoy the joke. Thank you for your time. Good luck on the final exam. If you have any questions, feel free to email me, and I will do my best to respond in a timely fashion. Talk to you soon. Bye.